Trying to start my video here. It's having a connection here. Well, I'm not getting my video to go. Just a minute here. There we go. Hi, are there any questions? Welcome to microbiology. I'm not hearing any questions, so let me show you the schedule. I guess I should show you my webpage too. I know you're not seeing anything. I'm logging into Canvas here. I'll show everything in a minute. Come on. All right, people are using the chat box. I don't use the chat box because when I'm sharing my screen, I can't see the chat box. Somebody has a really large message here. Are you ready for the quarter to begin? I'm ready. You can talk to each other, obviously. But anyways, I will not use the chat box. So if you have a question, don't send it in the chat box. I won't see it. Ask in the class. And I remember when I was first learning about Zoom, they suggested that 2 teachers run these Zoom sessions. So one person can put in emoticons and check the chat box and respond in the chat box. And then one person lectures. And of course, you've only got one instructor. So I'm going to warn you, I do not use the chat box because I won't see it when I am showing you my screen. Let me show you the screen now. All right, so you should be seeing my screen. This is the web page. Click on. Oops, went too far. What did I do here? Oh, well, that's interesting. That's an extra credit assignment that you can do for extra credit. I'll talk about that once I show you the website. So this is the Canvas website. Could somebody confirm that you can hear me and you're seeing the screen?
Yes.
Thank you.
Yeah, we can hear you.
All right, great. Yeah, am I loud enough?
Yes. Yes.
Great. All right, there's something you should read. There's the syllabus, which I'm going to show you and talk about in just a minute. Something for getting help to modify your Canvas site, so you make sure you're getting announcements from me. All of our class will be live, but I am recording it. And if you don't see that it's being recorded, I have to stop the share to be certain I'm recording there. So right there, I'm saying that we are recording it. And if ever you see it's not being recorded and I'm lecturing, remind me that we need to record it so that I can post it on YouTube. You can get to my YouTube site by clicking this link and open another window. It doesn't have any classes. Firefox can't open this page, so click on that YouTube site above. It won't open in Canvas, that's why it wouldn't open there. And then you'll see my link. Now these are classes from last term that we will put up classes from this term after we make them. Any questions about any of that? All right. They'll have captions on them. So I'll try and get them up after the lab today. Actually, I'm leaving right after the lab today, so I don't know if I'll get the lab up, but I'll try and get the lecture up during the lab. So you can watch them online if you're not able to attend the live session. This class is online and live via Zoom, meaning it's online only, for those of you who have questions. And if you're watching it at YouTube, meaning you're doing it after the live session, you cannot ask questions. It's the main advantage to coming to the live session during Zoom. It's also a little bit less interactive, but you can ask questions via email if you watch it over YouTube. Of course, in the live session, you can ask the questions. There's some YouTube videos from lecture animations. I've got links to them in our lecture site, but there's some of them. Biosafety information for students and physicians. This isn't terribly important because our lab is going to be online, so you won't be exposed to microorganisms. Something about the lockdown browser. You do need to take quizzes as well as the final in this class, and they will be online. And you have to use lockdown browser. So there's a little bit of information about that. And then the first 3 chapters of the textbook are right here, as well as the summary of all the chapters. We do not have all of the textbook online. You do need to go and buy the book, but if you don't have it yet, you can read the first 3 chapters on this.
Is it okay if we use like the 3rd edition, or do we have to have the 4th edition?
You can use earlier editions of the textbook, but you will have to figure out what the reading is. Is the bookstore having the 3rd custom edition of the let me see, the textbook that's custom made for Clark College? Have they got
Oh,
it?
I purchased it used on Facebook from a previous student.
Well, that's the 3rd edition. That's what I think I have for, yeah, the 3rd custom edition. That's the current one. I've got the textbook pages listed for the 3rd custom edition. Okay, so if you use an earlier edition, then you would have to figure out what the page numbers are, but I tell you what the reading is. And for earlier editions, you just have to figure out what it covers and then what the pages will be. Any question about that? All right, so let me go ahead and talk about that. In the syllabus at the very end, I've got the schedule. So for today, week 1, January 4th, we're going to cover technology session, the syllabus, tips for success. That's all covered in the syllabus. And then we'll get into chapter 1. And as part of chapter 1, which we won't cover today, but we will on Thursday, cover a little bit of chapter 10, the classification of microorganisms. And that, if you have the current edition of the textbook, 260, that's page 264 to 272, which I think that's not of the current edition of the textbook. If you get the full textbook, that will be the 12th edition of the textbook. I think the 13th edition is the current one out, but I believe the bookstore has the 12th edition because that's what we're using if you buy the full edition. The custom edition only has the chapters that we cover. The full edition has all the chapters in the textbook. Any question about any of that? All right. On the right side of the schedule, I'll have the lab, and I'll talk about that later in the day. Any questions about what we're covering? All right, if not, let me return to the web page. You do need to take the plagiarism tutorial and the plagiarism quiz, and you have to take the quiz by January 8th. That's this Saturday. It's worth 20 points. You have to get 100% on this plagiarism quiz, but you can take it something like 5 times. So if you don't succeed the first time, take it again, and if you need to, again. Very seldom do I have students have to do it more than 2 times. Most students complete it in 1 time. If you don't get 20 points, then there's an assignment. Let me see what that is. There's a 100-point assignment, which is the infectious disease research project. It's worth 100 points. If you don't get 20 points in the plagiarism quiz, then you get 0 points in the infectious disease research project. So make sure you take it again and again until you get 20 points on the plagiarism quiz. Any question about that? You do need to know about the citation style, APA citation style, class guide on that. That's what the plagiarism quiz will be talking to you about, as well as avoiding plagiarism and then citing something. So that's in the first block here. On the second block, I've got the week 1 lecture. Up there, we already have an overview and the objectives for chapter 1, and then the study guide. Let me show you a little bit about the study guide. The study guide gives you the lecture notes in a, what do we call that? Well, I'm going to say sentences. It's a little bit more than that. What is that called that I'm showing you here? Drawing a blank on what this is called, an outline format. And you can add your notes to it if you print it out. It follows what we're going to talk about, although occasionally the outline won't be 100%, meaning I may skip ahead. Actually, we do that one. Understand the beneficial role of microorganisms. Anyways, it might not be a perfect match with what we're covering, in which case you may need to skip ahead to add your notes and then back up to add your notes. But it is the material we're going to be covering, so you can add your notes there. I highly recommend you take notes, and when you're listening to the lessons, even if you're doing it on YouTube, I highly recommend you take notes, because if you just listen, you only learn about 10% of the material. And if you listen and take notes, because you're moving information from one part of the brain to another part to take the notes, you will actually retain about 20% of the material. And that's not very much, but it's twice listening to it alone. And I recommend you take notes and then go over the notes, review them before the quizzes and the final, as well as throughout the term. Because if you want to learn the material, do you know how many times you need to go over the material? Think about it. Like if you're playing a musical piece for the first time you play it, you're not going to have it memorized. Anyone have a guess for how many times you have to review something before you learn it? If you do, state it. Nobody has a guest, so I'll go on and say, you need to review the material about 20 times. And so if you take notes, you've done it twice, once from listening and then once from writing. And that's, of course, a lot shorter than 20 times. And this was mostly done with students in middle school or high school. And if you really want to learn the material and focus on it, you've got to review it at least 10 times. So you have to go over it time and time again. Any questions about that? And the best way to do it is to, of course, go over your own notes, because you've made these notes. You are, what do you call them, enlisted in it. Any question about any of that? The worksheet for lecture 1 is up. It's worth 2.5 points. We'll talk more about that. Extra credit. You can get extra credit by January 15th if you send me your picture. Nobody will ever see this. Make sure it's a picture of your face. I want to learn your names and I'm not very good at it. And if I see your smiling face with the grade book, when I'm adding your assignment grades, I will more easily learn your names. Okay. Another extra credit assignment is the extra credit study sheet. Let me click on that link here. It will get you a link to a spreadsheet, an Excel spreadsheet, and you can click on that link there. And what it is going to ask you for your schedule, your weekly schedule, what all you're doing. And if you think that's an invasion of your privacy, you can just generalize. Like you can put family time. You don't have to say bowling with the family or cooking, although that's not exactly an invasion of privacy. You don't have to tell me what you're cooking, just that you're cooking, and then dinner time. So put that on the first log. And then on the second log, this is your study schedule for when you're studying for microbiology. And if you use the same study schedule for week 1 as for all of the weeks for the term, you only need to do 1 log. But if week 1 you're studying differently, put it on log 2. And if week 2 it's different, put it on log 3. And just add logs if you need further weekly study schedules. And I'll compare your study schedule for what your schedule is and make sure you can do it. And then I will give you points for this. And what is that? 6 points. This is due, I think, Saturday of this week. And it's an tool to encourage you to figure out when you can study and how much you can study. You will get 3 points for your schedule and 3 points for your study schedule. And if you're not clear on the study schedule, you get a -1 penalty. So you only get 2 points out of that. But that's extra credit. So if you want some extra credit, go ahead and do that. Any questions about any of that? All right, let me move quickly through this.
Are there any other resources that you would recommend that we use besides like the textbook and the other stuff that you told us to buy?
Yeah, my syllabus goes through that. We'll go over that in a minute. And obviously the Canvas website also is good on giving you stuff to look at. All right, and then there's the week 1 lab. I'll talk more about that when we have the lab. So that's the assignment. I have all of the lecture up there, meaning it's all visible, but I don't want to overload you. So I only have the 4th week of the lab up and maybe some of the last of the week. Starting in week 5 of the lab, which is very long, and I'll talk a little bit about that. you're not seeing it because I don't want to overwhelm you with my website. All right, let's go to the syllabus. And I'll try and go through this quickly. We'll be always meeting from 5 o'clock to 6:10 or 6:15 for the lecture. I need a little bit of a downtime for the, the Zoom session to format and finish the lesson, so then I can upload it to YouTube. And if I only have 10 minutes, I won't have enough time for that to happen. And then the lab will start at 6:30. And I want to start at 6:30. And if the Zoom session hasn't finished what it's doing, it won't finish it when I start the next one. So we'll stop a little bit early. And if you see it's 6:15 and I haven't stopped, please let me know. And then we'll end the session and I will start the lab at 6:30. Contact information. I'm giving you my formal Clark email site, but I would prefer that you send me in the Canvas webpage. Let me show you that. There's the mailbox there. And if you send it in the Canvas email, I will check that at least every other day, meaning I will check it Monday through Friday, probably every day, and then about every other day for the weekends, meaning I won't check it every day in the weekend, but I will check the email frequently. If you send it to the Clark College email site, I will check it, but probably only twice a week. And that's because Clark College sends a lot of junk email, which I just try to delete. And so there's a lot of email I get and it's not important. So that's why I don't check it all the time. Any question about that? So if you want a quick response, send it to my Canvas email. And I've got an office, but I'm not going to be online. So it's best to email me. And for office hours, generally I'll try and talk to you either during the lab or after the lab so that we're both logged in. But we can arrange it online at some time. And after the lab, we'll just wait until everybody else logs off, and then you can have a private session. If it's something that you don't care about it being private, we can actually talk about it during the lab, because I will be there to answer questions. But once I've done telling you what to do and how to do it in the lab, I will be there to answer any questions. But you guys are going to be working on the lab, and so that's why we can talk during the lab. All right, the textbook and materials and other resources is provided right here. And you actually have 3 options for the textbook. Some optional text, they're not required, but they can help you. The photographic atlas is good for the lab. The microbiology coloring book, and then the textbook ready for microbiology is just good for studying. And then Canvas resources. The library, Course Reserve, has 3 copies of the textbook, a photographic atlas, and then 2 copies of the Get Ready for Microbiology. I don't know how they do that, but you can get those from the Course Reserve. I doubt it's an online book. I don't know if you have to, I don't think you have to go in, but you might only be able to get it for 2 hours. All right, Canvas will be a very good tool. It's your responsibility to visit Canvas and know about any updates I make. I'll send it by email, as well as make an announcement if there's a change in the schedule or another change in the class. And generally, we're an online class. So even if we get snowed in and colleges closes, we can still meet. Any questions about that? So I'm not anticipating any trouble. Course description, I'm going to ask you to read about that. Student course outcomes, policies and suggestions. If you're sick and you're not able to attend and you're not able to do the assignments, let me know. Contact me prior to any assignments are due, and then we'll make arrangements for a late work. You should read about preparation for the class. This class will be very busy. You will have a lot of material to do. You will have to review this material, and then you will have to make the assignments. I'll talk more about how much time this class requires in just a little bit. So don't wait to the end. If you fall behind and you need help, contact me early. Attendance is not going to be taken, so you may view the YouTube session. Like I said, the main advantage for doing it live is that students tend to remember the material better if you get it live, and you can also ask questions. The quizzes will be online. Most of them, you will need the lockdown browser to take the quizzes. And the quizzes are online, but you only have 24 hours to take the quiz. And if the quiz is missed, generally they cannot be made up. I know about the student responsibilities. I'm going to ask you to read that. You can read about the instructor responsibilities. I'll try and reply to your emails within 48 hours if you send to the Canvas email. Read about course withdrawal, late assignments, I'll mention that. The penalty for a late assignment is 25% reduction in grade for the first day it's late, and then 50% thereafter. I may not accept a late assignment if I've given the answers for that assignment. Generally, that doesn't happen, but, and also I don't accept late assignments during finals week. I'm sorry, I'm very busy during finals week. And so get your assignments in before assignment week if you turn them in late. You should know about the student conduct and what's expected of you. Generally, the only thing I'm going to mention is do not plagiarize. I do encourage you to work together, but you cannot simply put in your study buddy's answer. If you do, that's plagiarism. What you need to do is, if you want to use their exact words, is put it in quotes and say where you got it from. And that you will learn in the plagiarism tutorial. But you cannot obviously cheat. And if I find an assignment where you and somebody else have the same answer, even if it's just one question, you will get a 0 on that assignment. So you cannot plagiarize and you cannot cheat. Any question about that? Plagiarism is very important in school, and actually it is in life. And how important it is there was a sitting senator from Nebraska who in his graduate school plagiarized his either thesaurus or doctorate dissertation, and he was discovered on it, and he had to resign his Senate seat for plagiarism. So do not plagiarize. If you want to use somebody else's exact words, put it in quote and say where you got it from. Clark College has a policy now for reasonable accommodation for religious or conscientious, whatever, and read about that if you want to use it. Read about the etiquette for Zoom, suggested study methods. This is a challenging course. You will be learning a lot of vocabulary, a lot of new concepts. You will have several assignments due at the same time. If you're feeling stressed, that is normal for this class, but don't let it over stress you out. Some students actually find the lab their most enjoyable class in this class. So realize that you're taking on a very difficult class and a time hog class. You will need to be in class, either live or on Zoom or on YouTube, for about 3 hours of lecture per week and 4 hours of lab per week, for a total of 7 hours. And you will need to study about 14 hours for this class and lecture and lab in addition to that each week. And that's for an average student to get a B or an A grade. If you don't have the time, you might consider taking this class at another term when you have more time to study for this class. Any questions about that?
I do have a question regarding the lockdown browser. What exactly is that?
It is a software that you download, and you can take the quiz with this software. It's sort of a browser that locks you into the browser while you're taking the quiz, meaning you cannot get out of the browser while you're in the quiz.
Is there
Any
a time
questions?
limit for the, are there time limits?
Well,
on this?
there is a time limit for all of the quizzes. For the essay questions, you have to finish it within the day. So if you're starting at midnight, you have to get it finished by midnight. Submit your answers by midnight. For the other quizzes, there is a time limit for the multiple choice questions. And that's actually in Canvas, not by the lockdown browser, but the lockdown browser locks you into the browser, and that way you can't, I don't know, go on another browser and look up the answer online.
So is it like
Wait.
an app or is
What?
it a website? Is it an app or a website?
I've never downloaded it. I think it's a program and it's a browser.
Okay.
Okay. If you're taking the class with a phone, you might use the phone. It's not recommended because you'll not be able to access everything. But you could perhaps use it as an app. I do allow students to use, I think you call it a notebook for using the lockdown browser, but I would
Wait,
have to
so
look
the
it up.
quizzes are open note, right?
The quizzes are open note. However, if you look up answers, you will only be able to look up answers to 1 or 2 questions on the quiz because you will not have time to answer all the questions if you're spending time looking up the answers. Is that clear? That's why this is a timed exam, so that you cannot simply look up the answers.
But also
But
apply
yes,
to the
they
final,
are
like
open
open note
book.
as well.
What?
Does open note also apply to the final?
Yes. Meaning I can't stop you because this is an online class. You can obviously open up the textbook. Okay?
They're just making extra measures to where,
But I'm making it timed so that you can't look up all the answers. you will most be able to on a quiz, 1 or 2 answers. On the final, I've never figured out how many questions you can look up the answer to on the final. It's probably more than 2, but it's not terribly many. Maybe 4. It depends on how fast you answer the questions also. But yes, all of the quizzes that are multiple choice are timed and they use the lockdown browser. Now for the short answer and essay questions, you have 24 hours to get them done. And so in a sense, they're not exactly timed and you don't need to use the lockdown browser for them. And yes, you can look up those answers in the 24 hours. All right, so look at the list of suggestions for studying effectively. I don't have time to talk about that, but there's a lot of things you can do. Let me talk about the grading. These are the different assignments you can do. I want to speed up this, so I'm going to come down to the table. 20 points for the plagiarism quiz. We have 5 quizzes, but you get to drop your lowest quiz. So there's really only 4 quizzes that'll count at 75 points or 300 points. And on the quizzes, there's going to be a quiz A, which are essay questions, and quiz B, which are multiple choice. And I'm putting them together, A and B, to make one quiz. Lab assignments, there are 10 of them, although it's counting lab 00 and 01 as 1, 5 points each for 50 points. The identification of the unknown project, that's worth 75 points. There's the infectious disease project. These are both lab projects, by the way, 100 points. The lecture worksheets, there's about 10 of them at 2.5 points each or 25 points. And then there's the final 450 points. So that the total grade is out of 720 points. And there's about 20 extra credit assignment points. Okay, I've told you 2 extra credit assignments. There will be a third that I'll talk about next week. The course is not curved. So your points will be your earned points out of 720. The grading scale, if you get 92% or higher, you'll get an A. 90% or higher, you'll get an A minus. 88% or higher, or B plus. 82% or higher, or B, etc. We don't give a C minus, so 70% or higher, or C. And we don't give D minus or D plus, just a D. And then if you get less than 60%, you get an F. Canvas will tell you a grade, but it's using a scale slightly different than the scale here. So the grade in Canvas may be off, meaning the letter grade. So go with this percentage. You can read about the question review. I'll let you read about the microscope use. Support for students, here is a bunch of links which will give you some support, such as the Counseling and Health Center, the tutoring services, and yes, they do have online tutoring. And I think they have an online tutor in microbiology this term. I should warn you that some students feel that the online tutor in microbiology in previous terms didn't know very much. So if the tutor in microbiology tells you something funny, you should probably send me an email and ask me for support. Hopefully we'll get a better tutor. Help for disability and support services. The Canal Library, that's the Clark College Library, Computer Resource Center, and that is open, although I don't know why you would use that since it's an online class. And you can use it online. I don't know how you do that. And then help for students needing money for groceries, safe and stable place to live. So I'll let you read about that. I'll let you read about the non-discrimination policy that Clark College has. And then the school emergency procedures. Like I said, weather shouldn't affect us because we're online. I'll let you read about all that, but I don't think any of that applies to an online class. Safety information for students and physicians. This isn't terribly important because we're an online class, but you should know about the safety information because you're going to be likely working in the healthcare facility. Somebody sent me a chat. I don't access the chat, so hopefully you're talking to each other on the chat. And if you have a question, please ask it online. All right, I don't think I need to talk about that further. I should mention that Clark College has expectations for if you're on campus, and I've got this here because they asked me to add it to the syllabus. But for this class, we won't be meeting on campus. Words of advice and comfort. Sometimes it seems like your instructor is your enemy, and it is true, we do have to maintain standards. So sometimes we cannot accommodate you. However, you should know that we do want you, each and every one of you, to succeed. So I will do my best to work with you to get the work done. And if you have a problem, a complaint or concern, please let me know. Don't let it fester. It'll only get worse. Lab safety rules, you should learn about this for the lab, even though we won't be meeting in the lab, but you will be quizzed over it. And I'll talk about that when we have the lab. All right, the last page is the schedule. Is there any questions about the syllabus?
So we don't need to buy anything for the lab portion of this class, correct?
You do not need to buy a lab coat. What else do we require? It's been so long since I've actually been in the lab. Yeah, there's nothing you need to buy for the lab. And the lab coat, you didn't have to buy. You could use it from Clark College. Anyways, let me close that down. Well, no, I'll leave it up for the lab. Come down to the schedule. All right. If there's no further questions, let's move on into chapter 1. Chapter 1 is microbiology, the microbial world in you. And then in this lesson, we're going to cover a little bit of chapter 10, pages 272 to 281. And in the earlier edition of the textbook, and that would be the last edition before the current edition, page 264 to 272. Usually I'll have the first slide, a slide where I'm going to tell you what the major goals are and a rough outline. If you want all of the goals, meaning all of the objectives, you should come to my webpage. I'm looking for it, and see the objectives. On some of them, I have the objectives right with the study guide, meaning at the top of the study guide. I'm going to try and break that out and have overview and objectives as a separate link. And for chapter 1, I have it as a separate link. So all of the quiz questions, I'm having trouble accessing that because of there we go. Nope, that didn't work. Now I can go. Chapter 1, all of the objectives are listed here. And all of the quiz questions will come from an objective. Now, for the objectives, you have to know everything about that objective, meaning I can ask, like, what does the term pathogenic mean? I could ask that, but I could expect you to know what that term is and then say something like, oh, when is E. coli pathogenic? Or I could simply ask you, like, are most microorganisms pathogenic? That's in relation to the term pathogenic. My point is that, maybe I can get a better example. The process of fermentation. I could ask specifically about alcoholic fermentation or lactic acid fermentation. I could ask how many ATP are made during fermentation. I could ask a question like, is glycolysis a part of fermentation? All right, so you do need to know more than just what the one thing about fermentation. Any question about any of that? Anyways, if you know the objectives well, you should do well on the quizzes. Let's close that down. But I will give the major goals, meaning the most important objectives here. You should know the terms for this chapter. And we will talk about a brief history of microbiology. Probably that'll happen on Thursday. And then we will talk about the importance of microbes. And we're going to start actually with the importance of microbes, and we'll end talking about the importance of microbes. Any questions about what we're going to cover? All right. I should mention that if I lose the connection, that will sever my link with you, but I will restart the Zoom session. So stay logged in, and hopefully I will join back with Zoom. All right, let's talk about the importance of microbes. Here we're looking at the nitrogen cycle. Nitrogen can be said to start in the atmosphere. You can actually start at any place, but we're going to start it with the atmosphere. And some of the nitrogen can be fixed by bacteria and archaea and then made into an organic compound where the nitrogen's in an organic compound. Let's see if I can blow this up a little. And then the organic compounds can decompose when the cells die. Decomposition can happen by bacteria, archaea, and fungi, and then they'll be decomposing into ammonia, and some of that can be converted into ammonium ion. Green plants can use ammonia or ammonium, but they don't really like it. And if you were to put ammonia on your green plants, it would start looking sick and eventually would die if you were to get too much ammonia. Some of the organic compounds, this is trying to show you a minority, most of it will be decomposed into ammonia, but some of it can be taken up, the organic compound, by the green plants. And that would be like if the ammonia is put into a protein and then the protein's in the ground, the green plant could take it up. But most of the times that protein is going to be decomposed into ammonia, and then the green plants could take up the ammonia. Oops, went too far. The ammonia or ammonium can be converted by bacteria and archaea into nitrite. Nitrite can be converted into nitrate by bacteria. And nitrate is the form of ammonia that green plants really like. If you look at fertilizer for a green plant, you'll find that they'll have nitrate in there. Green plants love it. And then they can take that nitrogen, either the nitrate or the ammonia, and convert it into an organic compound, which can be decomposed mostly into ammonia, and a little bit can be decomposed into nitrate. And then the cycle continues. And then the nitrate can be denitrified by bacteria and archaea, converting the nitrate into nitrogen in the atmosphere. And our nitrogen cycle has now ended because it can continue. Any questions about that? How important are microbes in our lives and with the planet? Well, if we look at the steps that microorganisms are allowing that step to happen in the nitrogen cycle, it's shown wherever there's a red arrow. And you'll notice that almost all of the steps in the nitrogen cycle are happening because of microorganisms. Like this one right here and that one right there isn't happening because of microorganisms, but almost every other step is happening by microorganisms. Any question there? So this is showing you that the nitrogen cycle would not happen on our planet if there were not microorganisms, telling us how important microorganisms are. Otherwise, when something were to die, it would not be decomposed, and then the nitrogen could not be reused, and it would just be, it would no longer be available for use by the nitrogen cycle if there were no microorganisms. Any question about any of that? Microorganisms are also important in the generation of soil. Biogeochemistry, there are rock-eating microbes where they break down nutrients from rocks, leading to the formation of soils. Now, you should know that soil formation is not generally, the majority is not generally made by microorganisms. Mostly soil is made by erosion from wind and water. But microorganisms do make a significant proportion of the soil. So microbes are also important in making soil. Here we're looking at the level of oxygen in the Earth's atmosphere. We currently have about 21% oxygen in the Earth's atmosphere. And you'll notice when we go back in time, we have less and less oxygen until 2.7 billion years ago, we had no, or essentially no, oxygen in the Earth's atmosphere. Any question about any of that? So what started oxygen off in the Earth's atmosphere between 2.7 billion years ago and 1.8 billion years ago? Oxygen was made in the Earth's atmosphere by microorganisms. Shown here, that's the oxygen being made by a microorganism. This is cyanobacteria. And the oxygen being put in the Earth's atmosphere then allowed green plants to evolve. And we have 21% oxygen in the Earth's atmosphere because of the combination of microorganisms and green plants. So microorganisms were extremely important to get the process going. They did it entirely, getting the first oxygen in the Earth's atmosphere. And then even today, about 50% of the oxygen in the Earth's atmosphere is from the result of from microorganisms. And I guess I did the other 50% is from green plants. So microorganisms are extremely important in the generation of oxygen in the Earth's atmosphere. Any question about that? Here we have a sewage treatment plant. Humans are responsible for generating the waste whenever they flush their toilets or wash something down their drains, right? And humans break it down. That's why we built this right here. The structure puts in the waste and then breaks it down. Is that right? Come on, that's a yes or no question.
Yes.
Thank you for answering, but no, humans don't break it down. We build the facility for breaking it down, but we're not going in there and degrading the molecules in the waste, like when you flush your toilets. What happens is we have microorganisms which grow in this facility, and the microorganisms break down the waste, like all your toilet paper, as well as everything else. So in the sewage treatment plant, although we build the place for the waste to be broken down, but it's actually microorganisms which are breaking down the waste. And they'll turn it into CO2 and water, and then use the CO2 to make food. There is about 5% of the waste which will not be broken down. We're not talking about that. And then they'll just take that and then clean it. And I guess they turn it into soil. I don't know what they do with that. Let's talk about what microorganisms are. We use the term microbe to talk about microorganisms. I will be a little bit more specific, and when I'm talking about microorganisms, I mean something that has a cell and it's an organism, but we could use a virus as a microorganism, and I think your textbook does use it that way. It's not an organism because it doesn't have a cell, but we do call it a microbe because it's too small to be seen with the unaided eye. All microorganisms are single-celled. Microbes are either single-celled or smaller than a single cell, like a virus. They come in many shapes and sizes. Microbes include bacteria, archaea, some yeasts and all molds, protozoans, some algae, if it's a single-celled or single-celled, it's obviously a microbe. There is some algae which is much bigger than a microorganism, like the kelp that washes up on the Oregon and Washington beaches. That's not a microorganism. But some algae is a microorganism. All viruses and prions are microbes, and some people call them microorganisms. They're not really a microorganism because they don't have cell. So we can say they're special because they don't have cells. Microorganisms are essentially almost everywhere. So we can talk about them as the ubiquity of microorganisms, meaning we find them almost every place on the planet Earth. They play beneficial roles in numerous processes. I already mentioned the nitrogen cycle. Microorganisms are numerically the most abundant form of life on the planet. Now I've talked about a cell. Let's talk about what is a cell. A cell is the basic structural and functional unit of all organisms. It's a highly organized compartment bounded by a thin, flexible structure that we call the plasma membrane. And all organisms are composed of cells, unless you're calling a virus an organism, and a virus is too small to be a cell. And it does not have the component of a plasma membrane. You can find three things in a cell. One, I've already mentioned, the plasma membrane, and that establishes the boundary of that cell, what's inside and what's outside. Inside a cell, you can also find cytoplasm and DNA. The DNA, of course, coding for the genetic instructions for the organism. But cells always have cell membrane or a plasma membrane, cytoplasm, and DNA. In this slide, we're seeing different pictures of the planet, a pond with scum and plant life, the ocean with air above it and water. I'm not sure what you want to call this one. The ground, the surface of the ground, into the ground, the soil, a person and a cat, as well as the wall, the desert with the mountain and sand. I don't think that's a sand dune. I think that's a mountain. And then a volcanic vent with lava, steam, so there must be water coming in, and then rocks around it. So where on these pictures do we not find life? Do we find life everywhere in this picture of the pond?
Yes.
And I'm going to say within every inch. Yes, you find life everywhere, including in the pond water, on the scum, on the rocks. How about in this picture, the open ocean? Do we find life in the water and in the air above the ocean, open ocean? It's a yes or no question, people. Do you find life everywhere?
Yes.
Yes, you do. You do find it in the water, and you do find it in the air, especially spores, but also small cells can be blowing in the wind. How far up you can go into the atmosphere before you'll not find life is you need to go a mile up in the atmosphere before we don't have life. So you're going pretty high up and we still have life. And how about this picture? Do we see life everywhere?
Yes.
How about in the soil? Do we see life in the soil?
Not with the
Microorganisms?
naked eye.
I think you said probably yes, that's true. We find underneath the surface of the soil, we find life down about a mile. So you find life a mile above the surface and you find life down a mile into the surface. And for the ocean, that surface is the surface of the soil. So underneath the water, you can go down a mile underneath the surface of the ocean before you no longer have life. You'll find microorganisms down from 0 inches to a mile. Any questions about any of that? How about here? Do we find microorganisms on the cat,
Yes.
on the person's skin?
Yes.
How about the cat's fur and the person's sweater? It might be wool, it might not be, it doesn't really matter. Nobody's going to guess. You do find life everywhere in every inch of this picture, including the door or the wall. And how about in the the desert, would we find life in every inch of sand, every inch of the mountain, and every inch of the water?
Yes.
Yes, you do. Microorganisms. And you, once again, you go up a mile into the air before you no longer find microorganisms, or you go down a mile into the sand or soil. Okay, how about the volcanic vent? Is there any place you do not find life?
Very right into it.
Probably the only place you wouldn't would be inside the lava in particular, because it's too hot.
Yes, where you have the lava, either on the surface of the lava or in the lava, or I guess below the lava, immediately below the lava, you would not find living organisms because the heat would destroy the living organisms. But everywhere else, on the rock, the vent, rock above the vent, and the snow, there is living organisms in those areas. Somebody was trying to ask a question, but I couldn't hear you.
Would a tardigrade survive that?
I would not survive the lava. It would be too hot.
Because
And
I know in deep sea they're found around volcanic vents.
They're found around the volcanic vent. They're not found in the lava. Okay, so the highest temperature you can find life surviving in, it's something like a little over 120 degrees Celsius, something like 123 degrees Celsius. And that's actually not a tardigrade. You could find an endospore at that temperature. Tardigrade, I don't know what the highest temperature it can survive at, but it is found in the deep sea vents. But it can't take the temperature of the molten lava. But it could take of the boiling water in the vent, for example. All right, so where is everywhere? We mentioned that microorganisms are essentially everywhere. Everywhere is environments on the land and in the water. You can find microorganisms in the atmosphere. And in the atmosphere, you can go about a mile up and still find microorganisms. And under the land and under the water, you can go down about a mile and find microorganisms. In some of these, you can find plants and animals, but to be on the extreme, you would find microorganisms. You can find microorganisms essentially whenever water is present. You do need liquid water for life to at least metabolize, and that would be a temperature between -20, 120 degrees Celsius. Remember, 0 degrees Celsius is the temperature of frozen water. And we can find life alive down to a -20. But below that, we have not found any bacteria or archaea that will be metabolically active at more than a -20, something like a -40, for example. And it's just too cold, the cells would freeze. And how they survive a -20 is they have antifreeze in their cytoplasm. All right, microorganisms are numerically the most abundant life form on the planet. In 1 gram of soil, we have about 1 × 10⁸ microorganisms. And this will depend on the soil conditions, obviously. You could have less if you were in a dry region, but you could have more than 10⁸ if you were in a wet region and there was lots of nutrients around for the microorganisms to use. In a millimeter of ocean water, that's also 1 gram, would you have more or less microorganisms than 10⁸? That's a more or less question.
More.
Less?
That's a good guess. So thank you for answering, but no, you actually have less in the ocean. The deep ocean is actually one of the deserts on the planet as far as life is concerned. And we find less life in the deep ocean as well as the deserts than anywhere else on the planet. How about a gram of human feces? Do we have more or less microorganisms?
More.
We do have more as long as the human feces is fresh. And for the entire planet Earth, of course, we have more than we can find in a gram of soil. So this slide is showing you how many more or how many less. So in a gram of soil, 10 to the 8th microorganisms. In a gram of water, we only have 1 times 10 to the 6th microorganisms. In human feces, if the feces is very fresh, meaning just came out, we would have, sorry, I had to do a count here. We'd have 1 times 10 to the 11th microorganisms. But if it's been sitting out and dried up a little bit, you'd only have 1 times 10 to the 6th microorganisms in the human feces. Any question about any of that? All right, I already mentioned in soil, the numbers can vary. It's usually around 1 times 10 to the 8th. But if the soil is very dry, it doesn't have much carbon and nitrogen sources, it may be less than 10 to the 8th. And if it's very rich in these sources, carbon and nitrogen, you'll have lots of nitrate made or generated, and so we can have more. A human feces, as I mentioned, 1 times 10 to the 6th in a dry human feces, but in a fresh one, we have up to 1 times 10 to the 11th. And actually human feces, about 55% of the wet weight is from the weight of the microbes in the human feces. So the majority of the feces is actually microorganisms, at least by wet weight. On a human, we have many, many more than we do in a gram of ocean water. And here we're seeing on a human, we have about 3.8 times 10 to the 13 microorganisms living on and in the human being. Oftentimes these bacteria are symbiotic with us, meaning they don't harm us, but they do allow the Korean food to be what am I trying to say? You find microorganisms on the food, like at a Korean market, as well as any market where the food is not sterilized. In the entire Earth, we have 1 million microorganisms on the planet. That's estimated to be 1 times 10 to the 30th. So you have 1 here, and then 30 zeros following that number. A huge number of microorganisms on the planet. For humans, we only have, well, it's closer now to 8 billion people. It's a little less than 8 billion humans on the planet currently. Any question about any of that? All right. If there's no further questions, I think this is a good time to end. And so I'll start here next time, and I will see you in the lab for 6:30. Questions? If not, I'm going to log off.
Thank you.
All right.